Today, quite timely, we'll talk about various infectious diseases and epidemiology. So first, let's discuss what epidemiology studies. Epidemiology studies the spread of the disease in the population, the causes of the disease, the routes of transmission, the outcomes of the infection. Basically, these people are really important right now, considering the coronavirus pandemic. So there are two terms here, epidemic and pandemic. So epidemic is the disease that affects not just many, but more than usual at once. Usually, epidemics are characterized by very fast spread, and epidemic levels of transmission, epidemic number of cases is way higher than the regular number of cases for this disease. We observe it now for coronavirus. Influenza epidemics happen often. So Spanish flu was an epidemic, yellow fever in the late 19th century in Louisiana. AIDS is an epidemic. At some point, smallpox caused epidemics. Pandemic, not just a very large number of people. The entire globe is affected. Geographic area is extremely wide. So epidemiologists, they look at the disease process, and then they suggest where disease came from, so origin, and who are the people at the highest risk. Okay. Once epidemiologists have the hypothesis about this, they will do a controlled study, and they will analyze the results of that study. Three main methods of epidemiology are case studies, case control studies, and cohort studies. Also, epidemiologists do outbreak investigations, and what is super important about epidemiology is that the results of the studies have to be clearly communicated to health officials and to medical professionals. So what is the case study? So it's an analysis of the disease experiences for a patient or group of related patients. So for instance, you can do a case study of one coronavirus patient, or you can do a case study of a family and transmission within the family. So it's not universal, it's not uniform, but it allows scientists to formulate the hypothesis about the treatment or cause of the disease. And that is the main outcome of the case study. Now, another type is case control study. In case control study, the patients with a similar histories are observed, and they are collected from the original population. So patients all are from the same population. The disease is in the same stage. The whole question about how they possibly contracted the disease. And it's important to make sure that case control study is somehow linked with the original case study. The main outcome of case control study is to understand the route of transmission or mode of infection. And once the method of how disease is spread is identified, scientists can run a cohort study. A cohort study takes participants from the infected area, say from Ohio, and people that are enrolled in the study can be healthy or, no symptoms, or can have disease. But the important thing about cohort, they all should have a common element. So look at a particular cohort. coronavirus in teachers, coronavirus in students, coronavirus in healthcare workers. Okay? So the idea of cohort study is to understand the risk of people within a certain group, certain population to get sick. So this is what epidemiologists do now in regards to coronavirus. And the original appearance of coronavirus in the state of Washington, we called it an outbreak because it was sudden appearance in a small group of people. Now, obviously, epidemiology is not a perfect science. Sometimes it is difficult to select proper groups of people because people have different physiology, different lifestyle, different socioeconomic level. You have to take it into account that people who are more affluent may receive better health care, they may live in the better sanitary environments. And you can compare studies done in one country, but it becomes harder to compare studies done in countries with different lifestyle, for instance. Now, the main sources of error in epidemiological studies, they include, first of all, random error. Obviously, the population is variable. People can be of different age, smokers, non-smokers. Systemic error, problems with the data collection, okay? Or human error, technicians not correctly record the data. And then there's selection bias. Obviously, we see it with the coronavirus when people who get tested, they already have symptoms usually. So we do not expand testing on people who are asymptomatic. So that's a great example of the selection bias. Now, the international organization that coordinates epidemiological efforts in different countries, it's World Health Organization. It's a part of the United Nations. So this organization issues recommendations and guidance to how combat the epidemic, coordinates efforts of different scientists in order to provide vaccines to underprivileged groups of people, medical care to underprivileged group of peoples. And it's important that this organization is properly run and is under the effective leadership. So there are some epidemics. There's just a short list of it. Outbreaks of Rift Valley fever in Africa, known very well. Monkeypox in Brazil, Nipah virus in India, HIV is running rampant around the world. Every year we have flu. No more smallpox. Luckily, polio. No more polio. Measles, yes. Not epidemics, but outbreaks. Tuberculosis worldwide. Plague and leprosy are kind of rare. But yeah, it is correct. We should wash our hands thoroughly and often. Now, what happens during the disease? So first, pathogen has to enter the human host. That's what starts the disease. When it enters and changes the physiology of the organism, your immune system may overreact, or microorganism can produce some products that are toxic for human cells. So route of transmission can be different for different infections. It can be physical contact, like in case of Ebola virus. It can be inhalation of airborne particles, tuberculosis or coronavirus, or a direct injection of pathogen in the body. Malaria, microorganism, Plasmodium falciparum, is injected by a mosquito. And HIV, use of contaminated needles leads to blood-to-blood transmission. Now, when pathogen is in the body, it can start producing toxins. For instance, like bacteria can produce toxins, Staphylococcus aureus. When it contaminates the food, it produces the toxin that's responsible for food poisoning. Okay. Also, age is extremely important in determining the course of the disease. For instance, if a person is young, their immune system may not develop significantly enough to protect them from the disease, or they may not have enough stamina to survive through the disease process. And the elderly also, often they have immune responses that are slower or not as efficient, so that puts them at a particularly higher risk disease. So first group of microorganisms that we're going to talk about are bacteria. So the total number of bacterial species on Earth is enormous. We're talking billions. Only few of those actually can cause human disease. In this picture, you can see the comparison of a typical human cell versus E. coli. You see E. coli is much smaller. So this is a bacteria. This is a coccus, circular bacteria here. So they're prokaryotic, meaning that they do not have a nucleus. They can be found virtually everywhere. They're found in Antarctic. They do not have any internal partitioning, so no membranes. They do not have, as I mentioned, they don't have nucleus. They don't have membrane-bound organelles. They do have cytoplasm, cell membrane, DNA, and ribosome. So here you can see a typical structure of a bacterial cell. So membrane, you see plasma membrane and a cell wall sometimes, DNA, which is not in the nucleus, cytoplasm, those tiny dots are ribosomes, sometimes capsule, sometimes it can be flagellum for movement. But you see no nucleus, no internal membranes. And shape-wise, bacteria can generally be divided into several groups. I'm going to move here and then go back. So they can be circular cocci. If those circular bacteria form chains, we call them streptococci. If they form clumps, staphylococci. Bacilli, rod-shaped. Sometimes the curved rods are called vibrio. And this kind of a spiral bacteria called spirochetes. Bacteria are generally classified by shape, as we mentioned, and by staining. So for different microbes, if you stain them to see them under the microscope, bacteria that are gram-positive, they purple or red. Due to the gram stain. For example, Staphylococcus aureus and Streptococcus are red. Gram-negative bacteria are blue. Oh, sorry. Gram-positive bacteria are blue, yes, and gram-negative are red. Sorry. So to make sure that we got it. So gram-positive bacteria are blue and gram-negative bacteria are red. The difference between the two is that they have different structures of the cell walls. This is why they colored differently. So these are two classifications, like general classifications of bacteria. Now we live with an enormous number of bacteria, 2,000 different species in the gut, on the skin, in the mouth, reproductive organs, respiratory tract. And we also take antibiotics. And the problem with antibiotics, well, antibiotics are great. They treat infections, but antibiotics, they do not have very much deliberation between different bacteria. So antibiotics can target and inhibit protein synthesis. Other antibiotics can inhibit metabolic pathways. So some block synthesis of DNA and RNA, many break down the cell wall. So they have a variety of actions, but they're not very, very deliberate. And the problem is that bacteria can become resistant to antibiotics. Okay, they may not allow them to get into the cell, or antibiotic cannot attach to the surface. Bacterial metabolism changes and antibiotic is pumped out of the cell, or antibiotic can be destroyed. The problem is that all these mechanisms make bacteria resistant to antibiotics. And how can we make sure that they don't? Well, they will, but we have to make sure to minimize the development of antibiotic resistance. So we shouldn't use various antibacterial wash products. If we are prescribed antibiotics, we should take a full course, full allotment, all bacteria killed, and do not discard old antibiotics into water. So basically discard them properly.